Hello everybody, my name is Mark Temple. I'm a lecturer in molecular biology at Western Sydney University, and this is just a lecture discussion on colon cancer as a well-studied cancer, and then we'll introduce a couple of fairly generic concepts that are metastasis and angiogenesis. And the content and the narrative of the lecture is based on part of the chapter 11 of the Robert Weinberg book, on The Biology of Cancer. So one of the first ways of identifying cancer cells is by looking at tissue samples under a microscope. So typically cancer cells can be distinguished because they are different to the other cells in the particular sample. So this is showing a microscope slide taken at about 20 times magnification. It's looking at a structure called a crypt, and this is here are the normal crypts in this area of the slide, and highlighted here with this black arrow is a abnormal crypt. Okay, so this is referred to as a sort of an early stage cancer, an adenoma or an adenomatous crypt. So here's the some cells here which are distinct from the cells around. Now, typically these cells are less differentiated than the surrounding normal tissue cells, and it's the presence of these abnormal cells within the healthy tissue is one of the first signs that there's some abnormal cells growing in that tissue. So this is another look at what we've just been seeing in that slide, that 20 times slide. So this is we were looking at the top level. We're looking from this direction, from the top in the slide here. And if you look at what is actually going on here, this is the crypt. So this is like this region of cells here. Now, at the base of the crypt here are the stem cells, which are proliferating rapidly to give rise to these cells here, which move up through to the lining of the colon, and nutrients are absorbed through these cells, and these cells wear out and are constantly being replaced by new cells being made from these stem cells. Okay. So when we look at the progression of tumors, there's a simple concept which we consider, which is the multi-step tumorigenesis. So that is to say, tumors don't spontaneously occur overnight. They're not this. There's a long lead-up time and a long progression of changes accumulating in a population of cells. So the formation of a tumor is a complex process that usually proceeds over a period of time, typically a period of decades. And during this time frame, these normal cells begin to evolve into different classes of cells, and within that class of cells you get additional mutations, and you get the progression of more aggressive phenotypes leading to a fully blown cancer phenotype. Now, this kind of process is typically occurring in all of us, and it's happening in different parts of our body, but rarely does it ever reach a stage where it becomes clinically a problem. Okay, so there's slowly in our adult body cells, we're building up mutations over time, but rarely do we reach a stage where we're going to see a detectable tumor mass. So our bodies are quite resistant to the development of these cancers. Now, over this period of time, we have this multi-step tumorigenesis occurring, What's driving this process? Effectively, we're looking at mutations at the level of the genomic DNA, okay? Changes or alterations in the DNA content of cells. Now, these can be mutations, so a change in the base composition of DNA. Additionally, we can have epigenetic alterations that affect gene expression, such as methylation of DNA and other processes that we'll talk about in later lectures. Now, these alterations are occurring within the genome, but there are particular regions of the genome that are particularly sensitive to mutation. And these are in the regions of DNA that code for important proteins that control cell growth, cell proliferation, cell survival, cell differentiation. So there's a whole bunch of genes within the genome that the gene products of these, the proteins that are produced from these DNA sequences, are very important in controlling cell growth. Now, if these genes are mutated during this multi-step tumorigenesis over this long period of time in proliferating cells, then that will lead to small changes, giving those cells an advantage, a growth advantage, a replicative growth advantage of the surrounding, compared to the surrounding cells. Okay, so hence we get this malignant cell type, this malignant phenotype evolving in a population of cells that gives them a growth advantage. Okay, and this is driven by mutations in key genes that control cell survival and proliferation. So I've been saying to you that most cancers develop over many decades, and clearly this is based on long-term epidemiological studies where they've studied large populations of thousands of people over long periods of time, and then from this data they can inform us of characteristics of the development. Okay, the risk of a 10-year-old boy dying of colon cancer compared to that of a 70-year-old person, it's the 70-year-old is 1,000 times more likely to die of colon cancer than a young 10-year-old boy. So there's clear evidence that age is an important factor in the development of cancer, and hence the notion that it's an accumulation of events over a period of time. And we have some strong evidence in later slides that clearly show that it's an accumulation of mutations in a population of cells. So further evidence that human cancers develop over many decades of time come from looking at the incidence of lung cancer during the 20th century. Okay, so cigarette smoking was relatively uncommon early in the 20th century and then became more and more common as the century progressed. So during the wartime, soldiers were given cigarette rations. Soldiers went around the world and they spread the habit of smoking around the world. And in the 1970s, 20 years, 30 years after World War II, the rates of lung cancer were also increasing steeply. Okay, now cigarette smoking spread around the world, and currently annual deaths due to lung cancer caused by cigarette smoking exceed 1 million deaths annually, and this is thought to peak soon in 2020. So this is some data looking at the global consumption of cigarettes. So we're looking here, approximately here we have World War I, and here we have World War II, with some economic depressions as well, where people didn't buy as many cigarettes. And here you can see the spread of cigarette smoking across the 1960s, 70s, 80s, and 90s, with cigarette smoking increasing annually. So we're looking at 5,000 billion cigarettes being consumed annually within this time period here. So here we have a graph that shows the increase in cigarette smoking globally, and this parallels here with global lung cancer deaths. Okay, so if the midpoint of the peak here is, I don't know, 1960, 1970, the midpoint here is some, I don't know, 20 years later, so some decades later, you've got a parallel increase in the rates of lung cancer. So if you look at other non-tobacco related cancers and look at their increase over the same time frame, there's relatively little increase in the occurrence of cancers that are not associated with smoking. Okay, so here you have a cause, and here you have an effect suggested by this epidemiological evidence. And this indicates that there's a long period of time, many decades, whilst these multi-step processes are occurring in the cells to give rise to a cancer. But in these cases, it's a cancer that's a response to a carcinogen rather than a cancer that is an age-related cancer. So just to reiterate, when we're talking about this multi-step process, we're assuming that there's a sequence of unlikely events, meaning unlikely genetic mutations in key genes within the cells, and the build-up of these unlikely events lead to tumor formation. Now, these kind of events happen at comparable rates in our bodies. So we're considering here not a carcinogen. This is just general background mutations that occur in cells as a normal part of cell growth and development. So this kind of theory assumes that as we grow older, we will have completed some steps towards tumor formation, but not all of the steps towards tumor progression. Okay. So again, here we're looking at the incidence of cancer as they relate to age, and in this graph here, we're looking at age on the x-axis here, so younger children here, moving through to middle age, through to old age in this direction, from left to right. And on the y-axis, we're looking at the annual incidence of cancer per 100,000 males. Okay, so for instance, there's a purple line here which represents skin cancer. Okay, so here we have the rate of skin cancer increasing with age. So as you get older, the rate of annual incidence of skin cancer increases. Now clearly, skin cancer is age-related. We're not going to talk about the effects of ultraviolet radiation and how that drives skin cancer, and in parts of the world where it's high UV index, you get a lot more skin cancer. So it's just looking at generic effects. Okay. Again, we have stomach cancer. As you get older, the rate of stomach cancer increases. Okay. Here we have a dotted black line indicating the occurrence of prostate cancer. Again, young people, very low occurrence of prostate cancer. Older people, a high rate of prostate cancer. The rate here is more steep, and the different steepness of these graphs just indicates that we're looking at different cancers in different tissue types. And within each tissue type, there's a range of factors involved. There's a range of different mutations that are important in driving the cancer. So there are different rates, but in general, we're seeing this trend of it takes many decades to acquire enough mutations in a population of cells in a particular tissue to give rise to a particular type of cancer. So here's some further evidence to indicate that cancer incidence increases with age. Okay, so again here there's people have identified some large population studies from around the world, and it doesn't matter if you're in United States, United Kingdom, France, Norway. If you look at epithelial cancers, so cancers of the linings of the body, cells on the lining of the body, then clearly there's evidence that as you increase in age in either of these countries, then the rate of occurrence of cancer increases with age. Okay, so clearly this supports the multi-step hypothesis occurring over many decades of time. Okay, so cancer is not the instant transformation of normal cells into a cancer cell overnight. Overriding principle is that it takes many decades for these mutations to occur in populations of cells to give rise to a cancer. The other thing to consider when we look at this multi-step hypothesis over time is the fact that there are various carcinogens in the environment that cells can be exposed to, and repeated exposure to these carcinogens can lead to increases in the rate of cancer. So there was some really good evidence looking at mesothelioma, which is a cancer caused by asbestos, and there's some laboratory studies where they repeatedly apply a carcinogen to mice, leading to the formation of a tumor. Okay, and these are sort of related studies across different time frames, because mice live for much shorter periods of time than humans, but the same trends are observed through repeated exposure to carcinogens. And it becomes apparent from these studies, which support the multi-step hypothesis, that you need repeated exposure over extended periods of time. And it's the duration of the exposure rather than the absolute age of the individual that's important in driving the process. So we know age is a factor, but when you've got complications like these carcinogens in the environment, then we have cancers that relate to the time of exposure to the carcinogens. And there's some really good evidence here that demonstrates what I was just saying. So on the left here, we've got mesothelioma in response to asbestos exposure. And in the right here, we're looking at mice that are exposed to a carcinogen over repeated exposures. And what we're looking at here is the accumulation of either mesothelioma or accumulation of skin tumors due to the exposure of the mice. And if you look here, in both studies, we have age groups. So if we have the 15 to 24 year age group shown in blue, which is here, and then we have the 25 to 34 age group shown in red here, and we have the over 35s shown in green. Okay, and for either age group, okay, age is not a factor, because it's if you plot the data relative to the time of being first exposed, a 15-year-old after being exposed has the same rate of with these repeated exposures, has the same rate of accumulation leading to cancer as a 35-year-old. Okay, so we know that time and build up over decades is important, but when we look at a carcinogen in the environment, what we're saying here is that it's time after exposure that leads to cancer, and that's independent of age. And the same thing for mice. If you're looking at age of mice in weeks rather than years, because mice only live for a couple of years, then if you expose younger mice or older mice to the carcinogen at repeated and reproducible same kind of exposure times, then it's after the time of exposure you have the same rate of occurrence of the cancer. So again, this is saying that we're getting, over time, we're getting multiple hits occurring due to multiple exposures to the carcinogen, and this is further evidence of the multi-hit nature of cancer, requiring multiple mutations to occur in a group of cells, giving rise to a cancer.